So what I'm going to present here is picrust, and also how picrust feeds into a tool that's been published for a little while called STAMP. The idea by also showing STAMP is that it actually shows you what the data looks like and allows you to do some visualization. So for people that don't know me, I'm Morgan Langell. I just finished a postdoc and I started an assistant professor position in January at Dalhousie University in the Department of Pharmacology. I'm a Canada Research Chair candidate in human microbiomics. So let's just get started right away here. Okay, so I'll do the quick acknowledgements at the beginning, just in case we run out of time. So for picrust, picrust stands for Photogenic Investigation of Communities by Reconstruction of Unobserved States. Here's the URL. And I'm just one of many authors on the paper. So myself and Jesse were the two lead authors on it, but it involved quite a few other people just to bring together the package in a reasonable format. And then the other tool I just mentioned was STAMP. So STAMP is Statistical Analysis of Metagenomic Profiles. I don't have anything to do with STAMP myself. It was published by Donovan Parks and my previous postdoc supervisor, Rob Biko. And Donovan's still actively developing this, so I actually found a bug last night and he fixed it for me, so it wouldn't cause a problem with our tutorial today. And again, STAMP's really well documented, and I think you'll be impressed with sort of what you can do with that as well. So I'll just give you a brief overview of sort of what we're trying to do here. So in human microbiomics, there's kind of a couple of different approaches you can use to characterize a sample. One is you can use the 16S ribosomal RNA gene, and then you can process those sequences using a package like QIIME or possibly another, depending on what your preference is. And what you end up coming out of that is a table, an OTU table. So you have OTUs as row IDs here, and then you have some number of samples in the table as well. So if you had a small data set, you might have 10 samples or something, or you could have hundreds of samples as well. And then these OTU IDs are sometimes either represent an actual number or they're just sort of a filler from de novo OTU picking. And then obviously everything in the table is just an abundance for that OTU in each sample. And then from there you can do sort of lots of things, like you can build phylogenetic trees, you can do PCA analysis, you can do visualization over geographical space if you were doing anything that had actual geography coordinates. You can build networks, all that fun stuff. And then the other approach is shotgun metagenomics. So shotgun metagenomics is where you would take data and fragment all the different genomes. You would sequence hopefully most of the fragments, and you come up with information not just about sort of who was there, but also what they're doing. And then to annotate those reads, you would run something like, say, MGRAST, or Human, or Smash Community, or there's actually quite a few packages out there, all with advantages and disadvantages. And then from here, you end up sort of with a similar table to the OTUs. You come up with information about whatever your functional classification that you're looking at. So in most of the cases today, I'll be talking about KEGG orthologs, which look like these IDs. But often people use things like COG or NUG or PFAM or whatever your preference is. And then again, the same information can be used in similar ways to the OTUs, but you get information about functions as well. And then so what picrust basically does is it takes your OTU table here and basically gives you this without doing the sampling, without doing the sequencing. And so I'll just explain briefly how picrust sort of works. So the way picrust works is that you would have a reference 16S tree, and so what we usually use is the green genes tree, and this is just supposed to be a toy example. So the real green genes tree has hundreds of thousands of tips, And a lot of these tips, or most of these tips, are actually from environmental reads, so they don't actually, they represent organism, but we don't have a sequenced genome for it. And then from your actual microbiome sampling with 16S, you would get reads, and you can assign these reads to tips in this tree. And so that's what actually is called closed reference, OTU picking in CHIME, in the CHIME world. So you can associate a 16S sequence with each of these tips. So we'll just focus in now on this one, say, sub-branch. So now what picrust tries to do is it says, for this guy, for a particular gene, so we'll just focus on, say, one gene that we're interested in. We don't know what the representation is for that in green. But in pink here, or red, these are genomes that have been sequenced already. So we have a reference genome in a database, and the number is the copy number of that gene in that genome. So these guys just have 1, these guys have 1, this guy has 4, and this guy has 2. And what picrust does is it uses ancestral state reconstruction, and we tested a whole bunch of different methods to basically try to infer what the ancestor looked like based on these copy numbers. So you can use something like maximum parsimony, and there's lots of different maximum likelihood models as well to sort of model what this ancestral state looks like. And you get a plus minus around that as well. And then picrust basically takes this information and then propagates it down to the tip of the tree, and we get an estimate of what the copy number is in this genome. So that's for a single organism and for a single gene. So then if we're looking at, say, keg orthologs, which has, we're using a set of about 8,000 different genes, we actually have to repeat this for now every single gene in the keg ortholog. And then what we do to sort of speed things up when we use picrust is we pre-calculate what every single one of these tips looks like beforehand and provide the user with basically a database of pre-calculated tips in the tree. So for green genes, again, that's approximately 100,000. So that's how that's done. And then on the user end, what happens is that you give us an OTU table, and one restriction, so this is the actual input that you would give to picrust. So one restriction is that we require that you have green genes IDs as the observation IDs, or the OTU IDs. And this is a table that CHIME would give out when you do closed reference OTU picking. And also Mother has just added a feature now that creates biome tables that provides a green genes ID as well. And the reason we need this is that's how we actually reference those OTUs back to the green genes tree. Otherwise, we would have no idea how to associate them back to what we've already pre-calculated. Okay, so this is your input, and picrust does sort of two things. One, which is actually completely independent of predicting metagenomes and is actually quite useful anyway, is that we know that the 16S copy number varies between organisms. And it's mostly been sort of ignored up to this point, but you can imagine that it would have quite a drastic effect on the real organism abundances. So what we do is we actually estimate what the 16S copy number is for every single OTU, and then what we do is we normalize your OTU table based on this 16S copy number. And you can say, see, if you had this 1, 2, 3, 4, 5 OTU, if his copy number is 5, then it gets reduced to actual only being represented as twice in the table. So you come up with this normalized table that should better represent the real organism abundance in your OTU table. So then after that, we actually do the metagenome prediction step. So in the metagenome prediction step, we take this normalized table, although you could still use a regular OTU table as well, and we go into our pre-calculated large database here, and we simply are doing matrix multiplication where we multiply basically each of these KOs for each of these OTUs. So we know that with this OTU that these KOs belong to it, and so we collapse them based on these numbers into samples by KOs. And we get a metagenome prediction over here where now we have samples by the actual keg orthologs. Okay, so that's sort of how picrust works. Take questions, I guess, at the end. Is that the best idea?
Yeah, I think that's right.
Okay, great. So now I'm going to sort of move on to sort of the, I guess, the demo of it, and we can always come back to more questions after. So the brief tutorial that I'm going to walk through here is that I'm going to assume that you've already created an OTU table with either CHIME or mother, with the restriction that it has to have these green genes IDs. So as I mentioned already, this would be using closed reference picking, OTU picking, or in mother they've had this make biome file. So from there, I'm going to walk you through how we just correct for the 16S copy number, how we predict the metagenome, and how we collapse that to pathways. And then I'm going to show you STAMP, how we can visualize these OTU tables in STAMP, as well as the actual predictions coming out of picrust in here as well, and show you how STAMP can create things like PCA and correlations, and do lots of nice statistics for you as well. I should mention right before I get started that I'll be using sort of the Unix command line just because that's easier for me, and for a live demo that means it's locally on my computer. But picrust is located as well in a Galaxy interface on Curtis Huttenhauer's Galaxy instance. So here's the URL. And so if you don't want to have to install picrust or use it from a Unix terminal, you can run it through this Galaxy interface. So again, you would just upload the OTU table and then run through these different methods, and there's documentation as well to guide you through that. Okay, so let the fun begin. Okay, can people see what I'm typing here? Or is it too small? I can make it a little bit bigger.
No, it's fine.
It's fine? Okay. So, let me just see where I am here. Okay. So I know most people are going to not know how to use a terminal, I presume, Unix command line, but I'll just walk you through the major steps. And you could be able to do this yourself, or as I just mentioned, you could do this in Galaxy. So the two things we're going to start with is our OTU table. So that's here, otus.biome. And the other thing is we have a map file that describes the samples into different groups. So let me just show you the map file right now. So in this map file, this is just an example case. We have different samples, and so we have these as our sample IDs, so these can be anything. These have to match what's actually in the OTU table for sample IDs. And then after that, we have different groupings. You can imagine you could have like a disease state where we have healthy or sick that divides the samples up. Or in this case, these were actually taken from mice that have been aged. And so we have age groups for these different mice as well. So we have young, middle, and old. But you can have lots of different groupings as well. Okay, so we have our OTU table. So the first thing we're going to do is just normalize by the 16S copy number. And to do that, we simply type the normalize by copy number script. And so this assumes that you've installed picrust locally. And then we're going to output it to a different file. And that's going to just take a second to run. So this biome file now is going to exist. It's just another OTU table that's been normalized by the 16S copy number. Hopefully it's fairly quick here. Okay, so now that's done. So if we look now, we have a new OTU table as well. And then we're going to look at actually now making the predictions. So we're going to load in this corrected biome file. And output it to a different file. I should mention that I'll be doing KEGG orthologs, but it's also really easy. So that's the default. It's also really easy to get cloud predictions or RFRAM predictions, just using another option. And also, I think some people were interested in Nesty, so I'll put that as well right now. Okay, so this will take probably about a minute or two to run. So in the meantime, what I'm going to do is I'm going to start to show you what STAMP looks like. And we'll come back to this once it's finished. So STAMP is a Python GUI interface system. It's available for Windows, Mac, and Linux. In Windows, it's really actually dead simple. They have an installer with a thing that you would click on, and it's quite easy. For Macs, you have to sort of install from source, so you have to install the libraries that support it, and then usually have to launch it from the command line. But once you launch it, it's an interface that most people will like. Just a quick question, can you actually see this little window with yourselves in it on my screen, or is that not visible?
Yes, we can see ourselves.
Okay, so I'll just try to move that out of the way as much as possible. Or maybe I can just... Yeah, it's even better.
Okay. If maybe I can turn off our camera.
That's okay. I'll slide hide it over there. Okay, so the first thing you have to do is STAMP, of course, uses a different file format than the biome file. So we just have to convert our biome file into a sort of a plain text file that's readable by STAMP. So I'll just do that quickly right now. So we're going to load in. So I'll just show you what the OTUs look like. So we'll convert this corrected OTU file. From the metadata field, there's different selections. So we just choose that this is a Chime OTU table. And we save it as so. Okay, now that we've done that, we're actually going to load the data in. So STAMP takes two things. It takes the profile file, which could be OTUs or functions. It's basically just a table format. And then it also takes what I showed at the very first, this map file that indicates how you can group your samples. Okay, so I'll show you a couple things first. One is the group legend. Let me just pick this for a second. Okay, so the first thing you notice is that we have a group field, so this is where you can select now whatever your metadata, your mapping file contained. So you can choose disease state or age group, and when you select that, things are updated to now represent the samples that way. So I'm just going to use these three age groups to sort of demo this, and then I'll hide that and move it out of the way. The other thing I just want to show you briefly is the metadata table. So down here, this is the metadata table loaded in, and from down here you can actually deselect samples if you thought they were outliers or just to see what the data looked like without that one. And then you can also do lots of different filtering to filter your samples in different ways. We're just going to use all the samples, so I'm not going to show this anymore and move that out of the way as well. Okay, so the default for STAMP is to sort of create this PCA plot. And up here, this is important as well, is this profile level. So what these different levels represent are different levels in the Green Genes taxonomy. So level 7 is the lowest, and so these represent species. This would be genus, this would be family, class, phylum. Oh, did I miss one? Oh, sorry, order's in there as well. I'm just going to stick with family for right now at level 5. But if you want to see what this PCA plot looks like at different levels, it's really easy to change. So this is at the family level. If we go with more detailed information, at level 7, it's going to update and now change and show you this. And it colors the points by the grouping, and you can also sort of interact with this, so you can click on different samples to see what these guys represent. So it tells you the group that it's in and also the sample ID. Okay, so we'll stick with level 5. And then the other thing that you can do is, for the statistical side of things, is you can look over here. So you have choices of doing a multiple group statistical test, a two group test, or two samples. So for multiple groups, you have your different choices. So you can do ANOVA, which is your typical one, and then you can change these other parameters as well. You also have choice for multiple test correction, which is obviously always a good idea. So we can select this one for now. And then there's other options here about filtering based on the p-value or q-values that you get out of this. And this number of active features actually indicates how many categories are passing at that statistical test. So we'll just leave that settings for now. And then this drop-down box here lets you show your different visualizations. So if we go to a bar plot, it'll open up this window over here. And now you can see these are all the family OTU IDs. Here, just let me make this a bit bigger for a second. So you have your family ID, you have the effect size, you have your p-value and your corrected p-value. If we sort by corrected p-value, we're going to get our most significant things at the top. And then what happens is when you click on this, it's going to create a bar plot for that data. So I'm just going to move that over. This over. And so now we can see what the data looks like. So we can see why this p-value is created for the ANOVA. You can see the number of sequences. For the young group here is quite small. And then the middle and old group are significantly more. And then as you click through these different things, you can look to see what those plots look like. The other option is you can simplify it into a simple box plot. So instead of having individual samples, this is quite useful if you have a lot of samples and there's too many bars. So this is the same data just represented as a box plot. The heat map is relatively new, and there's some weird bug with Mac version, so I'm hoping that gets fixed really soon, but that's a nice feature. And then there's this other plot which does the group-wise comparison, but then shows you the pair-wise comparisons as well. And then this is the confidence interval, the difference in mean proportions. So the further to the right, the more significant, and then the different p-values for each group test as well. So that's group tests, and a brief walk through the plots. The other mention is that you can just compare two different groups. So say if we just wanted to compare young to middle here, again, there's lots of different statistical analysis, different t-tests, and multiple test correction as well. And then there's also the extra option now for a scatter plot, where you would have all the samples for the different OTUs, and you can click and get information about each of these OTUs as well, and about why is significant, as well as the overall correlation between these two samples, or two groups, I'm sorry. And then lastly, really quickly, you can do actual pairwise comparisons between any two samples if you're at that detailed level. And so you can do scatter plots as well as quite a few other plots as well. Okay, so that's kind of an overview of what STAMP does. I should mention, just for, I guess, the demo version, is that this Reconnaissance family, which we showed, was more representative in the middle and older. It was actually quite interesting. So there's a paper in Humans showing that Allostipes, which was a genus within this family, actually the only genus characterized, is overrepresented in older people as well. And so we're actually showing that this trend somewhat holds in mice, although it's kind of interesting that the middle group is higher than the old group. Okay, so that's how you can use STAMP for OTU analysis and visualization. Let's just go back to our predictions. It's finished. And now we have a couple of extra files. So now we have the KEGG predictions here. And we have this Nesty tab. Nesty contains information about the different samples. And so what this is, it's a metric of how representative your sample is compared to reference genomes. So basically what we do is that we have your OTUs, and we calculate the phylogenetic distance between every one of your OTUs to the nearest tip in the tree that has a sequenced genome. So you can imagine that the higher that phylogenetic distance is, the less our ability to predict would be. And then what we do is we actually weight it by the number of OTUs. So if your most dominant organism has a very distant genome, our predictions aren't going to be as good, and this will drive up the nesty value. Whereas if your most dominant organism is a very, very close neighbor in the tree, then they'll have more significance over things that are not as representative. So these numbers kind of indicate how well the prediction is thought to be across different samples. And I've found that's mostly useful if you have, say, a particular sample that seems to be an outlier in your data. You might be able to come in here and say, this guy has a nesty value quite a bit higher than some of the others. So maybe just the prediction for that one is not as good. That being said, there's lots of other different reasons going on besides nesty. And there's a plot in the original paper sort of showing the different nesty values compared to the environment that we're sampling. So for humans, this value seems to always be less than about 0.15. For mammals and sometimes soils, it ranges between 1.15 and 0.3, and then over 3, I would say those are very novel and diverse samples that probably won't be predicted very well with picrust. Okay, so last thing I want to show is, so these predictions from picrust are actual individual cake orthologs. And cake orthologs are good, but quite often we want to look at a higher level so we can look at the pathway level. And one of the complicated things about this is that one cake ortholog can be in more than one pathway. So that means collapsing them is not super straightforward, like we can collapse, say, OTUs into different taxonomy ranks. So what we're going to use is this categories by function script, which is included with picrust and is also on the Galaxy interface. And we're just going to give it the KO predictions. We have to give it the type of metadata that we're collapsing by. So if this was a cog table, you would collapse it by cog pathways. And we have to indicate the level. So usually 3 is the best, and we might be just changing this to the default, since after that it doesn't really matter as much. And we're going to just output this to a different file. Okay. So now we actually have our predictions. Yay, they're in biome format. And you can actually take these predictions, these functional predictions, and use them with lots of different tools, just like you would if you actually did shotgun metagenomics and you were going to use that data at the table format. So you can put these back into QIIME and do things with QIIME. You can convert these to a tab delimited file and do whatever your preference is, I guess. Again, I'm just going to show you how it works in Stamp World. So again, let me just really quickly convert this file. So here we're just going to pick cake pathways. Okay. So now we're going to load in that file along with the same map file that we had before. And an error, that's not good. I think it sometimes gives me an error when I'm already had data loaded in, but I think it's fine. So again, now we have different profile levels, just like we did with the OTUs. And so at level 3 is the actual pathway information. So now we can see over here, we have different pathways. We can do the same statistical testing we did with the, as with the OTUs. And again, we can rank these according to their corrected p-value. So we see here that at the very top we have steroid hormone biosynthesis. And again, we have, so we picked the group. So let's just change the group setting. So that was showing healthy and sick. Just going to change this back to age group. So it's actually more representative data. So we have at the top biosynthesis and biodegradation of secondary metabolites. And again, this is our box plot, again showing the number of sequences. And we can do the same sort of plots as before. So we can do a bar plot based on that. We can also do a PCA plot. So this is a PCA plot based on the functions. And as you know, this isn't really as informative as the OTUs, and that's kind of to be expected because it's well known that we often see more functional conservation as opposed to OTU variation, especially at the pathway level. So we get this type of information again, and we can look through these things and explore the data, just like we did with the OTUs, and we can change the statistical parameters. We can do two group tests, 2 sample tests. Really quickly, let me show you the raw K ortholog predictions as well. How am I doing for time?
Sorry?
Am I okay for time?
You are completely okay. Everyone is looking eagerly.
Okay.
And after that we are going for a coffee break anyway, so the break will just be a little bit shorter. That's
Okay.
all right.
Oh no, that's not good.
Oh, don't worry.
Okay, you can just see it calculating here. It's taking a little bit longer because there's actually 8,000 different KEGG orthologs, so the statistical test takes a little bit longer. But now you can see the individual KEGG IDs. I kind of have to work it out because this is obviously not super informative. You can look these up. You can just Google these and it's quite easy. We do have the description in the metadata field, so I'll probably be working on combining that so this is a little bit easier to interpret. But again, these are the individual KEGG ortholog predictions. We can do the same thing with our PCA plot. And we can rank these again according to their p-value. Oh shoot, the group always goes to the different thing. That's sorry. Good. Well, that's interesting. I have different KEGG orthologs. Oh, I must have done a correction yesterday. Okay, that doesn't matter. But again, we have the p-values here, and then we have different KEGG IDs as well. Let me just remove this. Sorry, in my demo I have different KEGG orthologs coming up as most significant, although that doesn't matter too much. Perfect, okay. So we'll assume that we have, these are most high-ranking, statistically significant KEGG orthologs. And the last thing I just want to show you is that, which is kind of interesting with picrust, is that since we have your OTU table, and we are making predictions on the functions, we actually have that direct link about who is contributing these functions. For this particular KEGG ortholog, we have the data to know how much of this is contributed by, say, a single OTU, or what's the diversity of the OTUs contributing to this different function. So to get that data, there's another script called metagenome contributions. Unfortunately, this is not up on the Galaxy interface yet. I should get around to adding this. But again, in here you would take your original OTU table. I forget how this is run. Copy and paste here. So we would have our original OTU table. here we would specify what K orthologs we're interested in. So you can leave it as default as nothing, but the table gets really, really huge. So the recommendation is kind of pick out a few K orthologs that you're interested in. So here we could pick out K01727. Right. Oh, now we're falling apart. So change windows, K01194. And so on. So you could list a few different ones here. Again, we would let this run. This takes a few minutes, so I'm going to sort of do the cooking show thing where they put it in the oven and take out the baked one. This just spits out a tab delimited file, and the format for it is a bit, it's not a standard, I guess, file format, but it isn't tab delimited, so you can open this in a Excel spreadsheet. And so I kind of went through this yesterday and just made it larger and a little bit cleaner format. So there are other columns in here, but I've just highlighted the most important ones. So what you get out from this metagenome contributions tab delimited file is the gene ID that we've sort of selected. And what I've done is I've just spread this into different spreadsheets for simplicity. Then you get the sample, and you get the OTU, and now we get the abundance information. So what this number means is that this OTU, with this green genes ID, contributed 244 of the functions associated with this in this sample. So what this information sort of tells you is that we saw in the in here that the middle age groups are ranging from, say, 200 to 400, depending on the different samples. And when we look at this table now, we can say that this OTU is contributing a lot of the signal across these different samples. This OTU is contributing some as well, and then as you go down, so these are just ordered by the amount of contribution, you see more diversity. Again, this file doesn't have a column yet for what this OTU represents, but this will be added shortly. What we'll have is another column here where you can actually see the green genes taxonomy for this OTU ID. And then at the same time, it'd be really nice to include the description of this cake ortholog, so that would be much nicer. But I just want to sort of demo, this is kind of what the output looks like. And you could do different visualizations with this as well. I think this is an area of development because we don't really have data like this much in the future, so I think it's an area where we still have to look at how we really represent this data well. So let's compare this with a different gene. So this is one of the other high-ranking genes. And you can see that this is really swamped now by the single OTU, meaning that it's just because we see this large increase in this single OTU, which, by the way, is a member of the Rickenelliaceae family that was significant before, that we're seeing a large contribution of the actual abundances for this particular one. And then lastly, this one's a bit more diverse, so we see more different contributions of different OTUs contributing to the reason for it being statistically significant. Okay, so I think that's the end of my demo.